So the topic is phenotyping as an indicator of genetic diversity in soybean. This is already the 20th talk of a series targeting to promote agrobiotechnology and enhance understanding of the potential applications of agricultural products. We hope that this series will inspire international scholars, researchers, farmers, and business in the agricultural field, as well as interested public. First, let me introduce our speaker, Ndiko Ludidi. He's a professor of biotechnology at the University of Western Cape. He's an established researcher with expertise in plant molecular biology, plant physiology, and biochemistry. He is a full-blood visiting research scholar at the University of Missouri in 2020. I think he just finished his trip to US and back to South Africa. So Professor Ludidi also served as a member of the management committee of the DSI NRF Center of Excellence in Food Security, where he is principal investigator in the Technological Innovation Program for the Climate Smart Regenerative Agricultural Project. So in the coming presentation, Professor Ndiko Ludidi will focus on comparative, qualitative, and quantitative assessment of soybean knolls as tools to determine agronomic traits in relation to yield and genetic determinants of resilience to environmental stresses. The environmental stresses that will be tackled are drought and high temperature. Now, so let's welcome Dr. Ludidi to the floor. So, Ndiko, please.
Thank you very much, Professor Lam. It's really a pleasure for me to make a contribution to this series. I must say that it's been quite a while since I have physically seen Prof. Lam and his group. We date quite a while back in terms of interaction on soybean as a crop and with the focus on research. So today I'm going to be talking about some of the work that I have done. So I'll try to share my screen. Sorry, let me just go back and try to get the screen that I want to share. I should have opened this presentation in advance, but it will be quick. Okay, so right. So in essence, my interest is broadly in how plants interact with their environment, but the focus being on interaction between plants and drought stress and also heat stress. So more part of the talk that I will have today will be focusing on that. I'm hoping that everybody can now see my screen. Just shout if you can't. As Prof. Lam has indicated, I am from the University of the Western Cape in Cape Town in South Africa. Just a little bit of background. Well, the background that I'm using for my slideshow today is actually from an area of South Africa within the province of the Eastern Cape. And this is a site where agricultural activity used to be quite prominent. So this is a portion of the fields where mainly maize has been grown, but a little bit of common bean has been grown in this area as well. And I think, you know, as a start to my talk, I would like to talk about soybean production in South Africa. And then we'll talk about some phenotypic characterization of some of the soybean accessions that I have access to, and then narrow that down to an investigation of some of the accessions in terms of their responses to drought. So if we were to ask where South Africa stands as far as soybean production, 2022 estimates based on cultivation or plantings in 2021 place South Africa as number 13. Let me see if I can get a pointer here. Okay, so you can see that in this list, there isn't any African country in the top producers until you get to South Africa at number 13. And the only other country that comes up within the top 21 producers, I would have said top 20, but I'm referring to top 21 here because Japan is a significant producer, but not so much of a producer, and yet quite a significant consumer of soybean. So South Africa stands at number 13, and the only other African country that appear here is Nigeria and Zambia. And of course, this means that these countries that appear here have a potential to be really significant producers of soybean. But if you look at how much they produce, it's really very little compared to Brazil, which is the top producer, and the United States. And in fact, top producers in the world would be Brazil, United States, and Argentina. China, despite being a major consumer of soybean products, is not the top producer. As a South African, I feel that we could produce much more than we are producing as a country because we have the capability to do so. But there, of course, there are some inhibiting circumstances that prevent South Africa from being one of the top producers of soybean in the world, but at least this one is the top producer of soybean in Africa. So that potential needs to be tapped. And if one looks at the growth rate of soy production globally, and this then speaks to the fact that there is potential for growth of soybean production across the world, except countries where this has been saturated, of course. So despite countries such as Egypt not appearing amongst the top producers, you can see that in terms of growth of soybean production, Egypt and Zambia as African countries show this potential of being able to increase soybean production much more, for example, than South Africa is doing. We're sitting at a growth rate of about 5%, while Egypt is 36% and Zambia is 27%. And I do wish that our country could emulate these two African countries, that is Egypt and Zambia, in terms of an increase in soybean production. Just to mention that despite the sort of marginal increase in China and in South Korea, the soybean accessions that I work with are from China and Korea. And of course, there are some South African accessions that I work with. So most of the talk that I will be talking about today would focus on accessions obtained from China, specifically Hong Kong through Prof. Lam's lab, and also some accessions that I've obtained from South Korea. And if one looks at soybean meal production, so this is the kind of primary or secondary product of soybean, because the grain is the primary product, which then gets processed through crushing to produce soybean meal, and that's the main agro processing product used for animal feed in various countries. Just to mention also that as much as many countries such as China and Japan use soybean mainly as a food source and also for other purposes, South Africa mainly uses soybean as animal feed. There is a small percentage that goes towards food consumption. And so that's why I do not mention these other products in the talk, but focus on soybean meal production. China, the United States, Brazil, and Argentina are top. And of course, this is a good thing in this because in terms of production of soybean grain, these are the top countries. And of course, South Africa sits way there at the bottom, but at least in the top 20. And so South Africa is a net importer of soybean meal, because as much as we are not necessarily a net importer of soybean grain, we are not processing enough of our soybean grain. And perhaps this points to opportunities for South Africa to build more soybean crushing plants as a way of processing the soybean that we produce in the country. So that of course doesn't speak to me as a plant scientist, but speaks to soybean producers and soybean processors. I am a budding soybean farmer, by the way, but my contribution is very negligible. And if one looks at the growth in terms of growth rate in the annual production of soybean meal, Cuba is really tapping into soybean meal production much more than any other country at the moment. And the growth rate the best growth rate in an African country is in Morocco. South Africa's growth rate is 6.35%, so around 6% compared to Morocco's meal production growth of 50%. So this tells us that South Africa really needs to work very hard to push that growth, and there's an opportunity for that. And that would really help in terms of increasing the variety of soybean products that can be produced in South Africa, which creates business opportunities that have a major impact on the economic sustainability of the country. So that needs to be tapped into. So in reality, what I could say is South Africa is sitting on a gold mine that it is not exploring. And for us to push towards growth of soybean production and soybean processing in the country, we need to identify varieties and even develop varieties of soybean that have the agronomic traits that are desirable for production within the South African context. Just by the way, I will mention today that within this field, this picture here is a picture taken a couple of years back when Professor Lam, who is standing here, visited this locality with me. So that's me there, and that's Professor Lam next to me. And these two budding soybean farmers, well, people that are interested in soybean farming. And the plan is to change this open field that is no longer in use into a soybean production field. So I think there's an opportunity for that. And I think if we can really expand production of soybean in fields such as this one, then we can make an impact in soybean production in South Africa. So now I'll talk about what we have been doing recently in terms of the work. So the first thing I will talk about is our efforts in phenotyping of the soybean varieties that we have. Excuse me, I just want to sort out something first. Just a moment, please. Excuse me about that. So we have tried to phenotype 19 varieties, well, let's say 19 accessions of soybean, some South African. I say South African because they were produced by normal conventional breeding in one of the agricultural research council's experimental stations in South Africa. And then, well, that was only 3 accessions. And the rest of the 16 accessions were accessions either from Prof. Lam's lab or from South Korea. And so the methodology that we used was really aimed at characterizing or phenotyping these plants when they reach the R8 stage of development. So this is the maturity stage, which is reflected here. So just to mention that before we did the phenotyping at R8 stage, even though I will not be showing the results of that today, we actually observed a very big variation in terms of the height of the plants as they go through the vegetative stages, the shapes of the leaves and the sizes of the leaves as well, but I'm not going to present that today. Just also to mention that the work was done in 2 sites in the Eastern Cape. So this is the Eastern Cape province of South Africa, and in a municipality, in 2 localities, in 2 municipalities. One in a site called Kolweni, which is not far from the slide background that I'm showing here, and one in a locality called Port St. John's, and that is approximately, give or take, about maybe 200 kilometers from this particular site that I say is shown in the slide background. So 2 sites over a period of 2 years. And what we did was to look at plant height and the first pod insertion. So the plant height is was simply a measurement of the length of the plant, or the height of the plant, by looking at the distance from the soil surface to the last node of the main stem, and per plot, because we had 1 accession per plot, we took randomly selected 12 plants to do the measurements. The first pod insertion was measured by looking at the length from the soil surface to the first pod of the main stem. This sometimes can be important because the location of the first pod from the ground can be a yield determinant, and it's actually important in cases where harvesting of the soybean is done using machines rather than hand, and also has implications in terms of proximity to the soil, because pods that are much closer to the soil tend to be contaminated by the soil and tend to have fungi contamination from the soil. So the higher up they are, the better. Then we looked at the time it takes for the soybean to reach full maturity. So our thinking here was that these factors would have, well, these phenotypic factors would reflect some aspect related to genetic differences between the genotypes that we had looked at. And we really wanted to find out which of these phenotypic or agronomic characteristics are related to genetic diversity. So in addition to these, we also looked at the weight of 1,000 seeds from each plot using 12 repetitions or replications based on 100 seeds. And then we multiply that by 10 in order to get the mass of 1,000 seeds. And this really reflects yield. And we thought that a reflection of yield could also be related to genetic diversity within the different genotypes that we're working with. Just to mention that we already saw some variation in the number of pods that we would have for each plant that would require to get 100 seeds. For some plants, just to get 100 seeds, we would require one plant because they would have sufficient pods to get 100 seeds from them. But some plants would require up to 6 plants to get 100 seeds. These would be plants that had few pods. So we would work with anything between 1 to 6 plants, because some plants would have as few as 18 pods and other plants would have so much pods that we would need only 2 plants. These would be plants that would have something like approximately 90 to 95 pods per plant. So we would need 2 plants there. So that's just a mistypo there. It should have been 95. And then we also determined yield based on an estimate of the kilograms of soybean seed weight per hectare. Of course, the plots were not a full hectare, but we could estimate this by multiplying the plot size. So in essence, what we did was to take plot size that are 4 by 4 meters by 4 meters, so 4 by 4 plots, and we would exclude the rest of the plots, about 0.5 meters up to 1 meter in the extremes, so that we focus only in the central part, which is the 4 by 4 meter part of the plot, because the plants in the extremities tend to vary a lot. So we're looking for consistency here. So from our analysis, we found that grain yield, which is the kilograms per hectare, was the most reflective characteristic in terms of the differences in these genotypes that we're looking at. So that contributed to about 92% in terms of the differences in grain yield. And the next most significant contributor was the height of the plant. Even though not shown here, I would also want to mention that aspect in terms of the number of pods. So if a farmer is looking at what genotype or what accession of variety they would want to adopt for growth or production or cultivation in these two sites. One of the things that we saw was instrumental in determining final yield was the number of pods per plant, which was closely related to grain yield. And also the taller the plants, the more branches they have, and therefore eventually the more pods they had. But that contributes only to about 5%. So the yield of the grain was the major determinant, and the one that was more related to the diversity of the accessions that we had. So having said that, we then screened these 19 cultivars in a greenhouse for their responses to drought and heat stress. And the reason really we had this focus on drought and heat stress is that South Africa is a semi-arid country, and therefore drought plays a major role as one of the stress factors that negatively impact crop production in South Africa. And of course, we know that as a result of climate change, much of South Africa has developed this pattern where droughts become more severe and they also become more frequent. So eventually we will have to try and come up with varieties that can withstand drought if we are to sustainably produce soybean in South Africa. And of course, in many cases, for soybean as a summer crop, in a semi-arid environment and with climate change leading to higher temperatures, it becomes important to incorporate heat stress in any research looking at tackling the impact of climate change on crop production. So that's why we focus on drought or water deficit stress and heat stress as well. So I would, just before I talk about this work, I just would want to specifically point out that this is a work of one of my PhD students, and that's Mr. Musa Akambi, who I believe is also in this talk. So this is really his work, and that work is facilitated by Mr. Lawrence Dingana, who is our research assistant based in the Eastern Cape. So he's the one who tends to the plants and make sure that they get enough water, and he removes the weeds. So I think it's important to just say that in advance. So when we screened these 19 cultivars or varieties or accessions of genotypes, we ended up selecting 2 of these, and this is because they are the most contrasting in terms of their responses to these stresses. So this particular genotype here versus this one, just by looking at the images here at the bottom, one can tell that you've got a lot of foliage under these conditions. So these are really optimal, well-watered conditions, and the plant is growing really nicely. Lots of pods on this plant. It's one of the accessions that also produces quite a lot of pods. It's in the range of the 90s. But under water deficit stress, the plant is still happy because it's, the leaves are not curling. You might not be able to see that here, but just visually, if you look at the number of pods, there's definitely less pods in this plant than in that one. So the drought stress has an impact on the yield of the plant. Heat stress, not so much impact, but a combination between heat stress and the water deficit really has a major impact just on the vegetative parts of the plant in terms of the amount of foliage, but also the number of the pods. But by comparison, if you look at what happens with this cultivar or this genotype versus this particular genotype, same trend in the sense that these stresses have a negative impact on the growth and the yield of the plant, but it becomes more pronounced just if you look at the amount of foliage in this plant versus this one and the number of pods comparatively, then you will see that drought and heat stress have more impact on this particular genotype. Just to say, I mean, it doesn't really mean anything. So this is one of the genotypes that we obtained from Prof. Lam, and this is one of the genotypes that we obtained from our collaborator in South Korea. But anyway, so that's just a visual thing. It's not quantitative, it's qualitative, so it doesn't really help that much. But if we select just a few characteristics, for example, relative water content, which is a reflection of how much water is in the plant, well, in the plant tissue. So for this particular genotype, under normal conditions, you have full tegger and the relative water content is above 95%. It drops quite a bit to way below 80% under drought and also under heat stress. And this might be important because we know that under heat stress, you get higher levels of evapotranspiration, and so this is I think this is one of the reasons why we have a drop in relative water content under the heat stress, even though we had supplied sufficient water to this plant. And then under a combination of water deficit and heat stress, we get even a much more pronounced decrease in the relative water content for this particular genotype. But then with the second genotype, which we then regard as the most sensitive amongst the genotypes that we've screened, simply because under the same condition as this one, the relative water content under drought stress is lowered to an extent that is below 60%. So if you look at that one, it's about 73, 75%, while this one sits at about 57, up to between 57 and 60%. So this means that this particular genotype has less capability to retain water in its tissues under drought stress, while this one is more efficient in retaining water. No difference between the two genotypes under heat stress, but under combined stress, much more decrease in relative water content in KDP2 compared to HMC 201. As you can see, this relative water content is sitting at about between 45 and 50%, while in the case of HMC, looking at about 55 to 60%. So much more water loss in KDP2 than HMC 201. And this is also reflected in the shoot weights. So I'm not going to go into detail to go bar by bar again, but very clearly, much more impact under combined stress in KDP2 than HMC 201 when one looks at the weight of the shoots. An interesting thing is that for KDP2 under heat stress, the shoot weight seems not to be affected. So it seems like KDP2 is happy with heat stress as far as shoot growth is concerned. And that is actually kind of reflected here. As you can see, this particular plant here, if you look at the number, or well, not really the number, but just the concentration of the pods in this plant here versus this part here, very little difference, if any. While for this one here, it's quite clear that you have a lot of pods sitting there, and a couple there, and very, very few in that region there. So HMC 201, as far as grain yield is concerned, would appear to be negatively affected by heat stress, while KTP2 isn't. So that's an interesting aspect in terms of these responses. So we are really keenly interested in finding out what the genetic determinants of these differences between KTP2 and HMC 201 with regards to their drought tolerance and their heat tolerance are. And at present, we are trying to drill down to these genetic determinants by looking at differential protein expression between these two contrasting genotypes. As I said, they are contrasting because they respond very, very differently to drought stress alone or heat stress alone, or a combination of these two stresses. So that is currently what we are busy with, the proteomic profiling of the two genotypes, simply because they are the ones with the most contrasting responses to the stresses. Just to go back to the little bit that I talked about in terms of the number of pods, which is eventually related to yield. If one looks at, well-watered conditions, HMC 201 has about 80 pods, and then under water-deprived conditions, that decreases to about 50. The heat stress also decreases the number of pods, and under water deprivation, when combined with the heat stress, you have an even more pronounced decrease than the individual stresses alone. And so this is the total number of pods per plant, by the way. And if one looks at KDP2, under well-watered conditions, 72, number of pods 72, water deprived, about 50. So just already you can see that water deficit has a negative effect on both these genotypes, but that becomes more pronounced under the combination stress. So here you can see there's quite a significant decrease in both genotypes, but the decrease in HMC 201 is less pronounced than the decrease in KTP2. So in reality, KTP2 is more sensitive to the combined stress than HMC 201. The work is ongoing in the sense that what we really want to understand is between these diverse genotypes, what is it that contributes to HMC 201 being much more capable of tolerating the drought stress than KTP2. And I think that's what I wanted to share with you today. And hopefully in the next presentation, I will share on the outcomes of our proteomic analysis that would link these phenotypic characterizations to which proteins get upregulated or downregulated in response to the stresses, but also which protein, how those proteins are linked to the genetic makeup of the individual plants, the individual accessions or genotypes. As a conclusion, of course, I would like to thank all of you for spending some time listening to the work that we have initiated, and of course, the work we initiated with Professor Lam. So this was years ago when Prof. Lam attended a conference in South Africa. He still looks the same in terms of aging. As you can see, I have a lot more gray hair and gray beard. So I have aged quite a lot since the last time Prof. Lam was here, but he has maintained his age very well compared to me. So I do not know what his secret is. I would like to know, of course. The work is funded by the National Research Foundation and the DSI NRF Center of Excellence in Food Security. Thank you very much. And of course, I acknowledge the contribution of all my students, but specifically Mr. Musa Agambi, whose PhD work this presentation is based on, and Mr. Sisipio Lawrence Dingana, who works in the experimental site in the Eastern Cape. Thank you. I will welcome questions, of course.
Thank you, Ndiko. So before we entertain questions, or I suppose we have a lot of questions about South Africa that Ndiko can answer. Well, I guess before we start, right? So before we start asking him questions, so I can see the one that I gave to Ndiko survived pretty well under heat and hot environment. So it's exciting because recently we also sent some of our lives to Pakistan, and it seems like that we have some heat-tolerant life. So maybe I start with my first question. So how important is heat-tolerant life for South Africa?
This should be really an important aspect, Prof. Lam, because what we have seen is that under extreme heat. So when I say extreme heat, you know, we in summer in South Africa we get these heat waves, and of course they have become worse. Essentially, we would get temperatures up between 40 and 42 degrees, and generally these used to be, you know, a few hours, and they would, at worst, they would last for 4 days. But we have had heat waves that last for 2 weeks. And so it's becoming worse. And what we've also noticed, of course, is that the heat has an impact during flowering, because we see that on hot days when the length of the the number of days of heat stress that occurs leads to some of the flowers actually abscissing, that is falling off from the plants. That has a negative impact. So the major soybean production regions in South Africa are more to the north, with the Free State and Mpumalanga being the major production areas in the country. And recently, we've had really severe heat and incidents of heat waves in both provinces. Mpumalanga is much warmer and more humid than the Free State. But in the recent years, there has been droughts combined with heat waves in both provinces. So this is really a problem. So it's quite important.
Okay, thank you. So let's see who's in the audience would like to know some more information from Ndiko. Just unmute yourself and talk. Joanna, you need to unmute yourself.
Dr. Ludidi, how are you?
Hi, Joanna. Good to see you again.
Yeah, same here. My question is, South Africa does have a significant production of soybeans. So are those mainly the genetically modified beans? That is my first question. And the number question is, how much of the soybean are used as food in South Africa? Like for consumption as food. Yeah, thank you.
All right. So, Joanna, the majority of South Africa's cultivated soybean in terms of production is actually reliant on large-scale farmer production. And about 90% of that is genetically modified, either for herbicide tolerance or insect pest resistance. So the majority of South African soybean is genetically modified. Having said that, though, small-scale farmers, even though they produce some of the genetically modified soybean simply because the seed market stocks most, well, the soybean stocked most by the seed market in South Africa is GMO, you will find that small-scale farmers also have a large majority of their soybean being non-GMO. So this is conventional soybean seeds supplied to small-scale farmers by the Agricultural Research Council, because the Agricultural Research Council has made their own collections of soybean, and they have breeding programs that focus mainly on yield and nematode resistance, based just on natural variation. So none of those are genetically modified. But with the commercial soybean producers, which account for the greater part of South Africa's soybean, those would be genetically modified. In terms of the amount of food, well, of consumption for food in South Africa, very little percentage. More than 90% of South Africa's soybean is channeled towards livestock feed, and then the rest goes to food consumption and the pharmaceuticals. And the pharmaceuticals from soybean in South Africa is very limited, almost really close to unestablished. But the food products would mainly be soya beans, but the soya beans is mainly really also for poor communities who cannot afford to buy their own meat. So they use soya beans then as a replacement for meat in their diets. But this is a small proportion compared to the use of soybean for livestock feed.
Thank you. So I saw a hand from Professor Sahya. So do you want to ask a question?
Yeah, thank you very much. Thank you. First of all, I congratulate you for this very useful presentation. I'm not a breeder, actually. I'm a microbiologist. I just want to have your comments that if we inoculate soybean with some thermotolerant bacterial strains, then what do you think about the tolerance, temperature tolerance in the soybean? We do have some pilot scale studies where we inoculated the soybean and we noticed that it performed better. So I need your comments on that.
Thank you very much, Professor. This is interesting to me. We haven't done anything from a microbial perspective in terms of testing plant beneficial microbes for heat stress tolerance, but I think it definitely would have an impact, and this is based on work we have done on alfalfa for salinity and also for responses to a fungal pathogen, former medicacenis. And we found that some of the endophytic bacterial isolates that we have actually protect these plants against salinity stress and also against the fungal pathogen. So I wouldn't be surprised if these, some of your isolates and perhaps then as a collaboration, some of our isolates could have a positive impact on heat stress. I'm not entirely sure of what would happen with drought stress. It would be nice to see what some of the inoculants, whether they are endophytic or rhizospheric bacteria, what their impact would be on drought. What we've seen from the work we've done in Medicago sativa was that we get more beneficial effects as far as salinity and disease responses are concerned, and less impact as far as drought is concerned. So it seems like, at least from our isolates, there is not a lot of protection against drought, but definitely against salinity and fungal pathogens. But we haven't really expanded this work a lot to get isolates from really arid regions. So perhaps if we could get isolates from hot arid regions, those isolates could have a protective effect on soybean. So that is one of the things. Yeah.
Thank you.
Okay. All right.
I think it's a very interesting idea to use biofertilizer, right? So like microbes to replace some of the fertilizers or to help the productivities under stress. This trend is very, very good, exciting. So hopefully you can see the success in South Africa. Professor Chan, do you have a question? I see you raise your hand.
Thank you, Professor Lam. My name is Long Chen. I'm from University of Western Australia. Thank you, Professor Ludidi, for your presentation today. Before I came to Australia, I saw the African countries probably are the most drought countries or continent there, but actually Australia is the drought continent or country in the world. In Western Australia and also in south part of Australia, the grain belt, or what we call the grow farms there, we are farmers, we're facing a serious problem of both drought and heat stress. So the combined both constraint is very important for us to look at how plants respond to that. I understand in northwest of China, farmers often use plastic mulching technology to prevent the soil moisture lost and also keep soil microbes. That lot of benefits for the environment also, but not plastic side anyway. Do you think then some technology or similar technology can be used in Africa? Can you comment on that? Thank you.
Thank you very much, Prof. Chen. I'm very pleased that you attended this talk as well. And of course, I've read the papers that you sent, and I'm very enthusiastic about working with somebody who looks at what happens below ground, because most of the work we do is above ground, and most of the activities that happen below ground are very, very important in determining what happens on the surface, of course. So you will be hearing a lot from me. I just want to say that before I respond to your question. But very importantly, yes, even though this wasn't part of my talk, there is evidence that some of the practices related to regenerative agriculture actually have a lot of benefit. For example, soil cover becomes quite important because that prevents water loss as a result of evaporation from the soil, and also has a protective impact on the microbes that live within the soil, and tends to reintroduce some of the beneficial microbes. So we have actually started with a project where one of my PhD students is looking at comparison between classical way, would say, modern ways of cultivation, soil tilling without any cover crops versus when we have cover crops. So we do this, we've started this this year with winter crops, and during summer we'll be switching to summer crops, and the summer crops we'll be looking at would be soybean as a legume, and maize and pearl millet as cereals. So I would probably be in a position to give an accurate answer in the first trial, at the end of the first trial, sometime in March next year. But I can already indicate to you that from a large-scale farmer point of view, this is seen as an impediment that requires a lot of input, but we've seen small-scale farmers doing these regenerative agriculture practices and with soil cover as part of it, and they have reaped benefits from it. And I think it's one of the things that we really have to adopt. When I was at the University of Missouri, we started talking about this to try and promote regenerative agriculture practices. So we will, if we get the funding, we'll be engaging on a larger scale project in much of the Eastern Cape to implement these practices as a way of preserving soil health and also promoting crop yield by reducing water loss from the soil using cover crops and mulching and all of those practices. So I definitely would say that it can be implemented in much of Africa, because most of the producers are small scale producers who are actually willing to find alternatives to these current conventional agricultural practices that the large scale farmers are using. So the answer is
thank
yes.
you. Yeah, thank you very much.
Good.
Thank you. So I follow up on that. So what's the prospect of intercropping? Because now I think you have a crop rotation. So how about intercropping with other economic crops?
Yes, the prospects are good, Prof. Lam. Yes, in South Africa, we have crop rotation between, for example, maize and soybean. But after some discussions with some of the small scale farmers, because I tend to talk more to the small scale farmers than the large scale farmers, because the large scale farmers, they kind of feel they should stick to their practices because it's been working for them. But I've spoken to one large scale farmer, and they are very willing to adopt intercropping. When I presented to them what we found in our small plots in Lukolweni, where Mr. Tingana works, because we intercropped maize and soybean, and also did an intercropping of pearl millet and soybean, and we really got good results. It's all about timing. To start with, what we did was to start with soybean and then follow on with maize when the soybean had grown enough not to be affected by the shade that would otherwise come as the maize grows. And we've got really, really good results. But it's still early stages because we've done that just for one season. So we want to do it in this coming season. So our season starts in November for the summer crops. So we'll be more able to tell what the impact is. But definitely, I think there are prospects for intercropping, and there's a lot of benefits associated with intercropping than waiting for 1 year to 2 years to change the crops.
Thank you. Okay, any other questions from the audience? Anyone want to know more about South Africa?
So I have seen a lot of good data, And what about the exclusive right? So I'm a patent attorney, so I care very much about how your fruits, right? You mentioned about the farmer expert, and that's my particular space. And but I also very much care about your last couple of slides about the genotype. So I'm just wondering, how have you really, your laboratory, thought about, like, you know, your wonderful research result not being misappropriated by other people?
Yes, so that aspect, Frank, is very important. And at this stage, you know, these results, of course, are not public domain, well, except today, of course, that some of us now have seen what some of our genotypes do. And of course, I think none of us here has access to those genotypes except those who collaborate with us. And of course, we have agreements as to how these genotypes can be used by who. But having said that, when it comes to us developing new genotypes that have specific characteristics like these ones that we've talked about, we have an intellectual property office that deals with these aspects. Of course, I am no expert in them. I'm just I'm just a plant physiologist. But our technology transfer office has a division that deals specifically with intellectual property. And before publishing anything and before presenting anything in a conference, we would consult with them so that they could, warn us if there is possibility of data being that is of intellectual property nature being taken off. And usually, I mean, I'm doing this in this particular case with specific genotype names. In publications, we simply code name the genotypes just to protect us in a case where we publish. But on the other hand, one can never be sure, and normally we consult with our technology transfer office that has this division. But of course, the patent lawyer like you working with Prof. Lam, well, it would be nice to have advice from time to time.
Yes. Anytime. Great. Thank you.
I think it's a related question, right? So to have IP rights, so it's very important to have your own breeding program. So so some of your collection right now is from us or from Korea. So I would very much like to see that your breeding program can thrive so that you have a new cultivar coming up that you can have a South Africa patent protection. So what is the status of breeding program of soybean in South Africa? Do you have lots of a big team or yeah? So can you explain a little bit?
Yes. So South Africa has very few qualified breeders, and this is now being addressed through the University of KwaZulu-Natal's program on crop breeding. So there is an effort to train a new crop of crop breeders. But just from our perspective, as my research group, we work closely with the Agricultural Research Council that has two people who focus on sorghum and maize breeding for the one person, and the one other person focuses on soybean breeding and cowpea breeding. So we have these two people that we work with, and my desire is to, in the next few years, to really start pushing for integration of these two people as part of a soybean breeding program to generate new varieties with the desired characteristics. And the main focus in the breeding program would be on heat and drought stress, because that is one of the major impediments to soybean production in South Africa.
So another question is, what is is there any particular advantage of using non-GMO, or it doesn't matter because you're going to use it for animal feed mainly, or so because there's in some country they have interest to grow non-GMO, so they need some special variety that can tolerate to the environmental stresses, and yet they are non-GMO. So how about in your country, in South Africa?
So South Africa has a non-government, well, has non-governmental organizations that try to push towards less use of GMOs. And it's a controversial aspect because of the fact that most of our food production in the country is actually driven by the large companies that promote adoption of GMO crops, simply because of the perceived benefits of the use of GMOs. For example, less use of mechanical means to control weeds, for example, and an easier approach to controlling some of the insect pests that destroy the crops. So there is a very clear leaning towards the use of GMOs because of the benefits associated with GMO technology. But these non-governmental organizations that promote avoiding the use of GMOs creates a space where we could look at alternatives to GMOs, and that's part of our interest in regenerative agriculture or agroecology, where adoption of agroecological technologies or practices can have the same benefits in terms of suppression of weeds and elimination of non-beneficial pests, for example. So there is a move now to push towards more agroecology and regenerative agriculture as an alternative. And I think that will eventually become very important because it has other benefits. For example, the use of pesticides and the use of herbicides has negative impacts on the environment. So if we can avoid using those by adopting agroecological and regenerative agriculture practices, then we'll be in a better space as a country. And there's really a move towards going that direction.
All right, thank you. So is there any other questions from the audience? Okay, it seems quite quiet. So yeah, maybe we make use of this time to do some advertisement for the next talk. So Katrin, can you show the poster? Yes, Dale, I would like to introduce our next talk. Right, so today we are more concerned about Africa, but 2 months later we have a talk on Argentina. So that will be more regional soybean cultivation. But in the next month, there's a very unique talk because as a biologist, one important thing is to measure the quality of our plants so that we can estimate the yield or whatever nutrients inside. So this is very labor intensive when we do it by biochemical means. So Professor Jin Wu from Hong Kong University, he has been working on hyperspectral analysis. So by flying a drone through the field or forest, the camera can take wavelength that's outside the visible range, so that there's a lot of interpretation that can be done by non-destructive way. So you can know your yield or even some of the quality of your product by interpreting the images taken by the camera. So, but how can it be achieved, and what what kind of applications related to this hyperspectral remote sensing technology agro ecosystem, as Ndiko emphasized. So Professor Jin-Woo will be talking about that on July 20th. So you can register through this QR code, or we'll also send you the invitation later. But this is a very new technique that probably will also help to improve the soybean production by non-destructive means of measurement. Okay, so hope that I will see you there in July. Okay, and Ndiko have a safe trip back home. And with that, I thank everybody from attending this seminar, and we will see each other again. Okay, so good night and good morning, everyone. Bye-bye.
Thank you very much, Prof. Bye.
Yeah, have a safe flight home.
Thank you. Thank you, Prof.
Yeah, well, I survived with you to the US, Ndiko, or she's waiting for you at home.
She actually came to meet me when I was in Mpumalanga, so we together in Johannesburg.
I see. Just help me to say hello to her. Right,
I will do so. I will do so, problem. Yes,
yeah, I hope that I can see you soon, because if the border is open again, I think I will organize a trip to South Africa, especially in Eastern Cape, right, to see how the soybean is going there.
Yes, that would be nice. So let's hope the borders will open.
So will Albert come? Albert, will you join me?
I will join. I would lovely to join.
Okay, so maybe we can organize a tour to the field. Yeah. Okay, that's fine, right? Maybe 2023. Let's hope for the best.
Yes, definitely. All right, thank you. Goodbye.
Right, so good night, everybody. Bye-bye.